So here's something worth thinking about as you settle in tonight. You've been living inside the same body your entire life. Every single moment of every single day, it's been right there, doing its thing. And yet, if I asked you right now to explain why your stomach growls, or what actually happens when you get a bruise, or why you sometimes twitch right as you're falling asleep, that little jolt, you know the one, you'd probably hesitate. Maybe you'd piece together something half-remembered from a biology class, or something you read once. But the full picture? The real mechanical, beautiful, slightly weird explanation? Most of us don't have it. And that's not because you're not paying attention. It's because your body is, honestly, the most complicated thing you will ever encounter. More complicated than any bridge, any spacecraft, any computing system ever built. And the wildest part is that nobody designed it. It just sort of assembled itself over billions of years through trial and error on a scale that's hard to even wrap your mind around. So tonight, let's just walk through it. Slowly. No rush. We'll talk about how some of this actually works, the stuff happening inside you right now, as you lie there, breathing, blinking, maybe feeling your heartbeat if you pay attention to it. And we'll talk about how people figured it out, because that's a story in itself. For most of human history, we had almost no idea what was going on under our own skin. The inside of the body was a mystery, and the theories people came up with to explain it were, well, creative. Beautifully wrong sometimes. Confidently wrong often. And the path from those early guesses to what we actually know now is full of the kind of drama you wouldn't expect. Rivalries, banned books, stolen corpses, careers ruined, lives saved by accident. All of it. Let's start with something so basic you probably don't even think about it. Breathing. You're doing it right now. You've been doing it since the moment you were born, and you'll keep doing it without any conscious effort for the rest of your life. Your body handles roughly 20,000 breaths a day. Think about that. 20,000. And you don't have to remember a single one. So what's actually happening? When you inhale, your diaphragm, this dome-shaped sheet of muscle sitting right below your lungs, contracts and flattens out. At the same time, the muscles between your ribs, called intercostals, pull your rib cage up and outward. This creates more space inside your chest cavity. And because there's now more space, the pressure inside your lungs drops below the pressure of the air outside your body. Air flows in, the same way air rushes into a room when you open a window on a windy day. It's just pressure equalization. Physics. Nothing magical about it, and yet it keeps you alive every second of every day. Then you exhale, and your diaphragm relaxes back up into its dome shape. The rib cage settles. The space shrinks. Pressure goes up, and air flows out. That's it. That's the basic pump. In and out. Driven by one muscle you've probably never seen and couldn't point to on a diagram. But here's what's really going on at a deeper level. The air you breathe in is about 21% oxygen. The air you breathe out is about 16% oxygen. So your body is skimming off roughly a quarter of the available oxygen from every single breath. Where does it go? Down into your lungs, which aren't just two big balloons. They're more like two trees, made of increasingly tiny tubes that branch and branch and branch until they end in roughly 300 million tiny air sacs called alveoli. 300 million. If you could somehow unfold all of them and lay them flat, they'd cover an area roughly the size of a tennis court. That's the surface area packed inside your chest right now, and the walls of those alveoli are so thin. We're talking 1 cell thick. that oxygen can pass right through them into the tiny blood vessels, the capillaries, wrapped around the outside. Carbon dioxide passes the other direction, out of the blood and into the air sac, and you breathe it out. This gas exchange is happening across that entire tennis court of surface area, every single breath, and it takes about 3/4 of a second. Now, people didn't always understand this. For a very long time, the prevailing idea was that breathing existed to cool the heart. Seriously, Galen of Pergamon, the Greek physician who lived in the 2nd century, around 130 to 210 AD, was one of the most influential medical thinkers in history. His ideas dominated Western medicine for over 1,000 years. And Galen believed the heart was like a furnace, generating what he called innate heat, and the lungs existed basically to fan the flames and vent the smoke. The air you breathed in cooled the heart, and the air you breathed out carried away the sooty vapors. It sounds almost poetic, doesn't it? And in a weird way, it's not entirely wrong. There is a metabolic heat component, and you do expel waste gas. But the mechanism, the actual understanding of gas exchange and oxygen transport, was completely absent. And here's the thing about Galen. He was brilliant. He was a physician to Roman gladiators, which gave him unusual access to open wounds and exposed organs. He did animal dissections, mostly on Barbary macaques, because dissecting human bodies was forbidden in Rome at the time. And from those dissections, he built an incredibly detailed and incredibly wrong model of human anatomy that went essentially unchallenged for 1,400 years. 1,400 years. Imagine believing something about your own body for that long and having it be fundamentally mistaken. It wasn't until 1543 that someone really started to tear Galen's model apart, and that someone was Andreas Vesalius, a young anatomist from Brussels who was only 28 years old when he published one of the most important books in the history of science. It was called De Humani Corporis Fabrica, on the fabric of the human body, and it was a masterpiece. Vesalius had done something that Galen never could. He dissected actual human cadavers carefully, systematically, and he drew what he actually saw rather than what he expected to see based on Galen's writings, and what he saw didn't match. The human body was different from the animal bodies Galen had based his work on, sometimes in small ways, sometimes in very significant ones. Vesalius found more than 200 errors in Galen's anatomical descriptions. 200. And publishing that was an act of real courage, because Galen was essentially sacred text in medical schools across Europe. Questioning him was like questioning scripture. Vesalius's own teacher, Jacobus Silvius in Paris, was so furious about the book that he called Vesalius a madman and declared that the human body must have changed since Galen's time, rather than accept that Galen had been wrong. Think about that for a second. A professor of medicine arguing that human anatomy had physically changed over the centuries rather than admitting his hero made mistakes. That's how deeply entrenched these ideas were. But Vesalius's book won out, slowly, because it had something Galen's work didn't: accuracy. The illustrations, many of them attributed to John van Kalkar, a student of Titian's, were stunningly detailed. Skeletons posed in contemplative positions against Italian landscapes. Muscular figures peeling back their own skin to reveal the layers beneath. It was art and science fused together in a way that had never been done before, and it changed everything. It established the principle that you learn anatomy by looking at actual bodies, not by reading ancient authorities. Direct observation over received wisdom. That sounds obvious now, But in the 16th century, it was revolutionary. And yet, even Vesalius didn't fully understand what was happening with breathing, or with the blood, or with the heart. He corrected the structural anatomy. He showed what things looked like and where they were. But the functional questions, the why and how, those took longer. A lot longer. Take the heart, for example. You can feel yours right now if you put your hand on your chest. It's beating, probably somewhere around 60 to 80 times a minute if you're lying down and relaxed. Over the course of a day, that's about 100,000 beats. Over a lifetime, roughly 2.5 billion. And each beat is pushing blood, about 5 liters of it in your whole body, through a network of blood vessels that, if you laid them end to end, would stretch something like 60,000 miles. That's enough to wrap around the earth more than twice. But for most of history, people didn't understand that blood circulated at all. Galen's model said the body had 2 separate blood systems. One was centered on the liver, which he believed was where blood was actually made from the food you ate. This dark, venous blood flowed outward through the veins to nourish the body's tissues, and it was consumed there, used up, like fuel. The other system was centered on the heart, specifically the left side, where venous blood supposedly seeped through invisible pores in the wall between the heart's 2 ventricles, mixed with air from the lungs, and became a brighter, more refined substance called arterial blood, which carried the vital spirit through the arteries. In Galen's model, blood didn't circulate. It was manufactured, distributed, and consumed. A one-way trip. And those invisible pores in the wall of the heart, they don't exist. Vesalius himself struggled with this. He looked and couldn't find them, and in later editions of his book, he admitted as much, writing that the septum, the wall between the ventricles, was just as thick and dense as the rest of the heart wall, and he couldn't see how blood could pass through it. But he didn't take the next step. He didn't say Galen was wrong about blood flow itself. He just said, essentially, I can't figure out how this part works. The person who finally did figure it out, who put the whole picture together, was William Harvey, an English physician who published his findings in 1628 in a slim little book called Exercitatio Anatomica de Motu Cordis et Sanguinis in Animalibus, which translates to an anatomical exercise on the motion of the heart and blood in animals. Harvey had studied at the University of Padua in Italy, which was the leading medical school in Europe at the time, and he'd learned from Hieronymus Fabricius, who had discovered the valves in veins, but hadn't understood what they were for. And this is where it gets really interesting, because Harvey's breakthrough wasn't based on some fancy new technology or a lucky discovery. It was based on logic, on math, really. He did a simple calculation. He estimated how much blood the heart pumped with each beat, just a small amount, maybe 2 ounces, and then multiplied it by the number of beats per hour, and the number he got was enormous. Far more blood than the body could possibly manufacture from food in that time. The liver couldn't be making that much blood. It was physically impossible, which meant the blood couldn't be getting consumed at the edges of the body. It had to be coming back. It had to be going around in a circle. Circulation. The blood leaves the heart through the arteries, travels out to the tissues, and then returns through the veins, back to the heart, to be pumped out again, over and over. The same blood, reused, recycled, circulating. And those valves that Fabricius had found in the veins, they were one-way valves, ensuring that blood in the veins could only flow in one direction, back toward the heart. Harvey demonstrated this with a beautifully simple experiment. He'd press on a vein in someone's forearm, push the blood out of a section, and show that it could only refill from one direction. The valves wouldn't let it flow the other way. It was elegant. It was right. And it made Harvey deeply unpopular with a lot of his colleagues. His medical practice actually suffered after he published. People called him a crackpot. Some physicians refused to accept circulation for decades. But the evidence was too strong, and eventually, the old Galenic model collapsed under its own weight. There was one piece Harvey couldn't explain, though. He knew blood went from arteries to veins somehow, out in the tissues, but he couldn't see how. The connection was too small to see with the naked eye. It wasn't until 1661, 4 years after Harvey's death, that an Italian physician named Marcello Malpighi looked at frog lungs under one of the newly invented microscopes and saw them, the capillaries. Tiny, impossibly thin blood vessels connecting the smallest arteries to the smallest veins, the missing link in Harvey's circuit. And Malpighi saw something else too. He saw blood cells moving through those capillaries in single file, one after another, squeezing through passages barely wide enough to fit them. The whole system suddenly made sense, from the massive chambers of the heart down to these microscopic tunnels threaded through every tissue in your body. And that's what's happening inside you right now. Your heart is squeezing, pushing blood through arteries that branch into smaller and smaller vessels until they become capillaries so narrow that red blood cells have to deform, actually change shape, to pass through them. In those capillaries, oxygen hops off the hemoglobin molecules in your red blood cells and diffuses into the surrounding tissue, and carbon dioxide makes the return trip, loading onto the hemoglobin for the ride back to the lungs. Then the capillaries merge into larger veins. The blood flows back to the heart, gets pumped to the lungs, dumps the carbon dioxide, picks up fresh oxygen, returns to the heart, and gets pumped out again. One complete circuit takes about 60 seconds. Right now, as you're lying there, your blood is making that full loop roughly once a minute. But here's something most people don't realize about the heart itself. Something that's genuinely strange when you stop to think about it. Your heart generates its own electrical signals. It doesn't need your brain to tell it to beat. If you took a heart completely out of a body, surgeons do this during transplants, and kept it in the right conditions, it would keep beating on its own. The signal originates in a small cluster of specialized cells in the upper right chamber called the sinoatrial node, sometimes called the heart's natural pacemaker. These cells spontaneously generate an electrical impulse about 60 to 100 times a minute, and that impulse spreads across the heart muscle in a precise, coordinated wave that makes the chambers contract in exactly the right sequence. Upper chambers first, then lower chambers, squeeze and release, squeeze and release. The electrical system of the heart is its own self-contained network, and when something goes wrong with it, when the signals misfire or take the wrong path, that's when you get arrhythmias, those irregular heartbeats that range from harmless flutters to life-threatening events. And the story of how we learned to read those electrical signals, how a Dutch physician named Willem Einthoven built the first practical electrocardiograph in 1903 and won the Nobel Prize for it. That's a story worth telling too, because the machine he built was absolutely enormous. We're talking about a device that weighed around 600 pounds. It used a very thin silvered quartz string, thinner than a human hair, suspended between powerful electromagnets. When the tiny electrical currents from the heart traveled through the body and reached electrodes placed on the patient's hands and one foot, those currents would cause that delicate string to deflect, to wobble side to side by the tiniest amount, and Eindhoven rigged up an optical system, a light source, a shadow projection, so that the movements of that string could be magnified and recorded on a moving photographic plate. The whole contraption required 5 people to operate it, and the patient had to sit with their hands and 1 foot submerged in buckets of salt water, which served as the electrodes. Buckets of salt water. That was the cutting edge of cardiac diagnostics in 1903. And yet, and this is what's remarkable, the basic waveform that Eindhoven recorded, the shape of the electrical signal he captured, is essentially the same pattern that doctors read on an ECG today. He labeled the peaks and valleys of that waveform with the letters P, Q, R, S, and T, and those labels stuck. More than 120 years later, every cardiologist in the world still uses Eindhoven's lettering system. The P wave represents the electrical impulse spreading across the upper chambers, the atria. The big spike, the QRS complex. That's the powerful contraction of the ventricles, the lower chambers doing the heavy pumping. And the T wave is the ventricles resetting, repolarizing, getting ready for the next beat. Each of those little squiggles tells a story about what the heart is doing electrically, and when a doctor looks at an ECG printout and sees something off, a widened QRS, a missing P wave, an inverted T, they can often pinpoint exactly where in the heart's electrical system the problem lives. Think about what that means. Before Eindhoven, if your heart was misbehaving, a doctor could listen to it with a stethoscope, feel your pulse, and basically guess. After Eindhoven, they could see the electrical signature of each heartbeat drawn out on paper, a literal map of the signal as it traveled through the muscle. It was like going from hearing a car engine make a weird noise to actually being able to read the engine's diagnostic computer. That's the leap. But let's step back from the heart for a moment, because there's another system in your body that's doing something extraordinary right now, something so constant and so automatic that you've probably never given it a second thought tonight. Your kidneys. You've got 2 of them, tucked up against your back, just below your rib cage, each one about the size of your fist. And what they're doing right now is genuinely astonishing when you break it down. Every single minute, your kidneys filter about 1 liter of blood. Over the course of a day, that adds up to something like 150 liters, roughly 40 gallons, of fluid that gets filtered through these 2 small organs. Now, you obviously don't produce 40 gallons of urine a day. That would be a problem. What happens is that your kidneys filter almost everything out of the blood: water, salts, glucose, amino acids, waste products, all of it. And then they selectively reabsorb about 99% of it back into the bloodstream. They keep the good stuff and let the waste and excess water pass through as urine. So out of those 150 liters that get filtered, only about 1 to 2 liters actually leave your body. The functional unit of the kidney, the tiny structure that does this filtering, is called the nephron. And each kidney contains roughly 1 million of them. 1 million tiny filtration units, each one consisting of a little ball of capillaries called the glomerulus, wrapped in a cup-shaped structure called Bowman's capsule, connected to a long winding tubule where all that selective reabsorption happens. The blood enters the glomerulus under pressure, and the pressure forces water and small molecules through the capillary walls into Bowman's capsule, kind of like squeezing a wet sponge through a very fine sieve. Big things, blood cells, large proteins, can't fit through, so they stay in the blood. Everything else gets pushed into the tubule, and then as it flows along this winding tube, The cells lining the tubule walls actively pump useful molecules back into the surrounding blood vessels. Glucose gets reclaimed. Amino acids get reclaimed. Sodium and potassium get carefully balanced. The tubule cells are making millions of tiny decisions every second about what to keep and what to discard. And here's where it gets really clever. Your kidneys don't just filter waste. They regulate the entire chemical environment of your blood. If you drink a lot of water, your kidneys dilute your urine and get rid of the excess. If you're dehydrated, they concentrate it, pulling back almost every drop of water they can. If there's too much sodium in your blood, they dump the extra. Too little, and they hold on to it. They're constantly adjusting, constantly fine-tuning, maintaining this incredibly narrow range of conditions that your cells need to function. The pH of your blood, for instance, the acid-base balance, stays between about 7.35 and 7.45. That's an extraordinarily tight range. If it drifts outside that window by even a small amount, enzymes start malfunctioning, proteins change shape, and things go wrong fast. Your kidneys are one of the main systems keeping that number locked in place, and they do it without you ever being aware of it. The person who first figured out how the nephron actually works was an American named Homer Smith, working in the 1930s and 40s at New York University. Smith was one of those scientists who was absolutely obsessed with his subject. He spent decades studying kidney function, and he developed the clearance techniques, methods for measuring exactly how much of a given substance the kidneys were removing from the blood per unit of time. That became the foundation of renal physiology. Before Smith, people knew the kidneys made urine, but the details of how they filtered, what they reabsorbed, and how they regulated blood chemistry were murky at best. Smith brought mathematical rigor to it. He figured out that you could inject a substance called inulin into the bloodstream. Inulin is a plant sugar that gets filtered by the glomerulus, but isn't reabsorbed or secreted by the tubules at all. And by measuring how much inulin appeared in the urine, you could calculate exactly how much fluid the kidneys were filtering per minute. That measurement is called the glomerular filtration rate, or GFR, and it's still one of the most important numbers in kidney medicine today. When your doctor checks your kidney function with a blood test, they're essentially estimating your GFR. But anyway, the point is that your kidneys are doing all of this right now, silently, while you lie there. And they're doing it in coordination with hormones, with signals from your brain, with feedback from your blood vessels. It's a whole conversation happening inside you, a constant negotiation between systems, and that word, hormones, that opens up a whole other dimension of how your body works, one that took centuries to even begin to understand. Because hormones are chemical messengers, molecules released by one organ that travel through the blood and affect a completely different organ somewhere else in the body. And for a very long time, nobody had any idea this kind of long-distance chemical signaling was even possible. The breakthrough came in 1902 from two British physiologists named William Bayliss and Ernest Starling, working at University College London. They were studying digestion, specifically how the pancreas knows when to release its digestive juices into the small intestine. The prevailing theory at the time was that it was all controlled by nerves. The thinking was that when food entered the small intestine, nerve signals traveled to the pancreas and told it to start secreting. Bayliss and Starling tested this by cutting all the nerves to a section of small intestine in a dog, every single nerve fiber severed, and then introducing acid into that denervated section. If the nerve theory was right, nothing should happen because the communication lines had been cut. But the pancreas responded anyway. It started secreting just as vigorously as before. So Bayliss and Starling tried something else. They scraped the lining of the small intestine, ground it up, mixed it with acid, filtered it, and injected the resulting liquid into the dog's bloodstream. And the pancreas responded again. Something in the intestinal lining, some chemical substance, was being released into the blood and traveling to the pancreas, telling it to get to work. They called this substance secretin, and it was the first hormone ever identified. Starling later coined the word hormone itself, from the Greek word meaning to set in motion or to excite. And that discovery cracked open an entirely new field. Because once you knew that the body used chemical messengers traveling through the blood, you could start looking for them everywhere. And they were everywhere. The thyroid gland in your neck releases hormones that control your metabolic rate, how fast your cells burn energy. The adrenal glands sitting on top of your kidneys release cortisol when you're stressed and adrenaline when you're frightened. That sudden rush you feel when something startles you. The racing heart, the sharpened focus. That's adrenaline flooding your system in a fraction of a second. The pituitary gland, this tiny structure hanging from the base of your brain, barely the size of a pea, releases hormones that control growth, reproduction, and even how much water your kidneys reabsorb. It was once called the master gland because it seemed to orchestrate so many other glands, though we now know it takes its orders from the hypothalamus, a region of the brain just above it, which makes the whole system even more interconnected than anyone initially imagined. And this is where the body starts to feel less like a collection of separate organs and more like a network, a web of signals and responses where everything is talking to everything else. Your brain talks to your hypothalamus. Your hypothalamus talks to your pituitary. Your pituitary talks to your thyroid, your adrenals, your ovaries or testes, and those glands talk back, sending hormones into the blood that loop all the way up to the brain, telling it whether to increase or decrease the signal. It's a feedback loop, the same kind of control mechanism that engineers use in thermostats and autopilot systems. And the person who really mapped out these feedback loops in the endocrine system, who showed how the hypothalamus integrates signals from the nervous system and the hormonal system into one unified control network, was a Polish-born researcher named Andrew Shalley, working alongside Roger Gilliman in what became one of the most bitter scientific rivalries of the 20th century. Both of them were racing to isolate the tiny releasing hormones that the hypothalamus uses to control the pituitary, and the competition between them was absolutely ferocious. We're talking about two men who started out as colleagues, who actually worked in the same lab for a time under the same mentor, and who ended up in a decades-long race that consumed their careers, their labs, and honestly, their personal lives. It's one of those stories where the science is extraordinary, but the human drama is almost unbelievable. So let's back up just a little. By the mid-20th century, endocrinologists knew that the pituitary gland released all these powerful hormones: growth hormone, hormones that controlled the thyroid, hormones that triggered ovulation. But they had a nagging question. What tells the pituitary what to do? It couldn't just be firing blindly. Something had to be controlling it. And the leading idea, championed by a British anatomist named Geoffrey Harris in the 1950s, was that the hypothalamus, that small region of the brain sitting right above the pituitary, was sending chemical signals down through a special network of tiny blood vessels connecting the two. These chemical signals would be releasing factors, hormones that told the pituitary to release its own hormones. Harris couldn't isolate them, though. He could show that the connection between hypothalamus and pituitary mattered, that if you cut those tiny blood vessels, the pituitary stopped working properly, but he couldn't hold up a molecule and say, "Here, this is the signal." That's where Schally and Guillemin come in. Andrew Schally, born in Poland in 1926, survived the Second World War as a teenager, eventually made his way to England and then to North America. Roger Guillemin, born in France in 1924, trained as a physician in Lyon and then came to North America as well. Both ended up working with the same mentor in Montreal in the early 1950s, and both became obsessed with the same question. Could you actually isolate these hypothalamic releasing factors and prove they existed as real, identifiable molecules? They parted ways. Schally went to the Veterans Administration Hospital in New Orleans. Guillemin went to Baylor College of Medicine in Houston, and later to the Salk Institute in San Diego. And for the next 20 years, they essentially ran parallel research programs with the same goal, each desperately trying to beat the other to the finish line. The problem was staggering. These releasing hormones exist in unimaginably tiny quantities. The hypothalamus is already small, and the amount of releasing factor it contains is measured in micrograms, millionths of a gram. To get enough material to identify a single molecule, you needed thousands, sometimes hundreds of thousands, of animal hypothalami. Schally's lab was processing pig brains from slaughterhouses. Guillemin's lab was doing the same with sheep brains. We're talking about literally millions of brain fragments over the years, ground up, extracted, purified, tested, purified again in this grueling biochemical marathon. And the rivalry was personal. They stopped speaking to each other. They would publish papers that pointedly failed to cite the other's work. Their students and postdocs absorbed the animosity. Scientific conferences became tense when both were in the room. And here's the thing. The race wasn't just about ego, though ego was certainly part of it. It was about whether these releasing factors even existed as discrete molecules. Some very respected scientists thought Harris's hypothesis was wrong, that the hypothalamus controlled the pituitary through nerve signals, not chemical ones. So Schally and Guillemin were also racing against skepticism, trying to prove that the whole framework was correct. Guillemin's team got there first with one molecule, thyrotropin-releasing hormone, or TRH, which tells the pituitary to release thyroid-stimulating hormone. They reported its structure in 1969. But Schally's team published the same structure almost simultaneously, within weeks. Then Schally pulled ahead with the next big one, gonadotropin-releasing hormone, GnRH, which controls the pituitary hormones that drive reproduction. He reported its structure in 1971. Both men shared the Nobel Prize in Physiology or Medicine in 1977, along with Rosalind Yalow, who developed the radioimmunoassay technique that made measuring tiny hormone concentrations possible in the first place. And honestly, Yalow's contribution might be the most practically important of the three, because her technique is what allowed doctors everywhere to actually measure hormone levels in patients' blood, which transformed endocrinology from a somewhat speculative field into a precise quantitative one. But the point of this whole story, beyond the drama, is what it revealed about your body. Your hypothalamus sits there, This tiny region weighing maybe 4 grams, and it integrates information from all over your nervous system, your body temperature, your blood sugar, your stress levels, your sleep-wake cycle, signals from your gut about whether you've eaten, and it converts all of that neural information into hormonal commands. It's the place where your brain, your nervous system, and your endocrine system all merge into one thing. And the releasing hormones it sends to the pituitary are exquisitely precise. TRH is just 3 amino acids long. 3. That's barely a molecule by protein standards, and yet it controls your entire thyroid axis, which in turn controls your metabolic rate, your body temperature, how fast your heart beats at rest, how quickly you burn calories. Think about what that means. A 3 amino acid peptide, released in nanogram quantities from a region of your brain you've never consciously thought about, is setting the pace of your entire metabolism right now as you lie here. And this connects to something you've probably experienced without knowing the mechanism. Have you ever noticed how your body seems to know things before you consciously decide them? How you start sweating before you've registered that you're nervous? How your heart speeds up at the sound of a door slamming before you've had time to think about whether it's dangerous? That's because the hypothalamus and the autonomic nervous system, the part of your nervous system that runs without your conscious input, are constantly adjusting things behind the scenes. Your body is not waiting for you to make decisions. It's making them for you, millions of them, every hour. Let's talk about that autonomic nervous system for a moment, because it's one of the most elegant pieces of engineering in your body, and also one of the most misunderstood. People sometimes hear autonomic and think it means automatic, which is close enough. It runs automatically, without you having to think about it. But it's not a single system. It's 2 systems working in opposition, like 2 hands on a steering wheel pulling in different directions, and the balance between them determines your physiological state at any given moment. The sympathetic nervous system is your accelerator. It's the fight-or-flight system. When it activates, your heart rate goes up, your pupils dilate, your airways open wider, blood flow shifts away from your digestive system and toward your muscles. Your liver dumps glucose into your bloodstream for quick energy, and your adrenal glands release adrenaline and noradrenaline. Everything is geared toward action, toward dealing with a threat. The parasympathetic nervous system is your brake. It's sometimes called the rest and digest system. When it dominates, your heart rate slows, your pupils constrict, your digestive system becomes more active, your body shifts into maintenance mode, repairing tissues, storing energy, doing the housekeeping work that keeps you healthy long term. And right now, as you're lying here, hopefully feeling relaxed, your parasympathetic system is gradually taking over. Your heart rate is probably a little slower than it was an hour ago. Your breathing is deeper and more regular. Your digestive system, if you ate a few hours ago, is quietly working through whatever you had for dinner. This is your body shifting into its nighttime maintenance mode, and it's doing it without any input from you whatsoever. The person who first really mapped out this dual system was John Newport Langley, a Cambridge physiologist working around the turn of the 20th century. Langley spent years painstakingly tracing nerve fibers, applying nicotine and other drugs to nerve ganglia to see which ones blocked which responses, and he's the one who coined the terms autonomic nervous system, sympathetic, and parasympathetic in the framework we still use today. He published his comprehensive account in 1921, and it's remarkable how well his basic architecture has held up. The details have been refined enormously. we now know far more about the neurotransmitters involved, the receptor subtypes, the way the brain modulates autonomic output. But Langley's fundamental map of two opposing systems balancing each other is still the foundation. And here's what's really clever about the design. It's not that one system turns on and the other turns off. They're both active all the time, in varying degrees. Your heart, for example, receives input from both sympathetic and parasympathetic nerves simultaneously. The parasympathetic vagus nerve is constantly sending signals that slow your heart down, and sympathetic nerves are constantly sending signals that speed it up, and your actual heart rate at any given moment is the net result of those two competing influences. It's like having one foot on the gas and one on the brake at the same time, which sounds inefficient, but it actually gives you incredibly precise control. You can speed up or slow down almost instantly just by shifting the balance without having to start one system from scratch. And that vagus nerve, by the way, is worth pausing on because it's one of the most remarkable nerves in your entire body. The word vagus comes from the Latin for wandering, the same root as vagrant or vagabond, and it's called that because it wanders all over. It's the longest cranial nerve, running from your brainstem all the way down through your neck, into your chest, past your heart and lungs, and deep into your abdomen, reaching your stomach, intestines, liver, and other organs. It carries parasympathetic signals to almost all of your major organs. When you take a slow, deep breath and feel your heart rate drop slightly, that's the vagus nerve. When you feel that calming sensation after a big exhale, that slight settling of your whole system, vagus nerve. There's a reason that breathing exercises work for anxiety. Deep, slow breathing stimulates the vagus nerve, which increases parasympathetic tone, which tells your heart to slow down and your body to relax. You're literally hacking your own autonomic nervous system through the mechanical act of breathing, which, when you think about it, is one of the few places where the voluntary and involuntary parts of your nervous system overlap. You can't consciously slow your heart rate directly, but you can breathe slowly, and your vagus nerve will slow your heart rate for you. This interplay between conscious and unconscious control is something that fascinated early physiologists, and it connects to one of the deepest questions about how your body is organized. Because on one level, you are a single organism with a unified experience of being alive. But on another level, you're an astonishing collection of semi-autonomous systems, each running its own programs, each responding to its own signals, and most of them operating entirely beneath your awareness. Your immune system, for instance, is making life and death decisions right now, identifying potential threats, deciding which cells to attack and which to leave alone, and you have absolutely no conscious awareness of any of it. And the story of how we came to understand the immune system is, in some ways, even more dramatic than the endocrine story, because it forced scientists to completely rethink what it means for the body to know itself, to distinguish self from non-self. Because that's really what immunity comes down to, when you strip away all the complexity. Your body needs a way to tell the difference between things that belong inside you and things that don't, between your own cells and an invading bacterium, between a healthy lung cell and a virus that's hijacked it. And that sounds simple enough in principle, but when you start thinking about how you'd actually engineer a system like that, a system that can recognize millions of different threats it's never encountered before, while leaving your own trillion-plus cells completely unharmed, you start to realize just how extraordinary this problem is. For most of human history, people had no idea this system even existed. They knew, of course, that some people survived plagues and others didn't. They noticed that survivors of certain diseases seemed protected afterward. The ancient Greeks observed this during the plague of Athens in 430 BC. Thucydides wrote that those who had recovered from the disease could nurse the sick without catching it again, because, as he put it, the same man was never attacked twice. That's a remarkable observation. He didn't know why it happened, but he saw it clearly. And that basic insight, that surviving a disease can protect you from getting it again, sat there for over 2,000 years before anyone could explain the mechanism behind it. The first real breakthrough, and you've probably heard this story, came from Edward Jenner in 1796. Jenner was a country doctor in Gloucestershire, England, and he'd noticed something that local milkmaids had known for years. Women who caught cowpox, a relatively mild disease they picked up from infected cow udders, seemed to be protected against smallpox, which was one of the most devastating diseases in human history. Smallpox killed roughly 3 out of every 10 people who caught it, and those who survived were often left with terrible scarring or blindness. It was a horror. And here were these milkmaids walking around with clear skin during smallpox outbreaks, and the local folk wisdom was that cowpox had somehow shielded them. So Jenner did something that would be, let's say, ethically complicated by modern standards. He took material from a cowpox sore on the hand of a milkmaid named Sarah Nelmes and deliberately inoculated an 8-year-old boy named James Phipps. The boy developed a mild cowpox infection, recovered, and then, here's the critical part, Jenner later exposed him to actual smallpox material. And nothing happened. The boy was immune. Jenner repeated this with other subjects, published his findings in 1798, and coined the term vaccination from vacca, the Latin word for cow. And that's the origin of the word you've heard your whole life. But here's the thing that's easy to miss. Jenner had no idea why this worked. None. He couldn't tell you what was happening inside James Phipps's body that made him resistant to smallpox. He just knew that it worked, empirically, observationally. The mechanism, the actual engineering of immunity, remained completely opaque. And it would stay opaque for almost another century, until a fierce debate erupted in the late 1800s that would split the scientific world into 2 camps, each convinced the other was wrong, and both of them, as it turned out, partly right. On one side, you had the cellularists, led by a brilliant, eccentric Russian zoologist named Elie Metchnikoff. In 1882, Metchnikoff was living in Messina, Sicily, working on starfish larvae, about as far from human medicine as you can imagine. And he did this beautiful, simple experiment. He took a tiny thorn from a tangerine tree and poked it into a transparent starfish larva, then watched under a microscope. What he saw changed everything. Within hours, mobile cells swarmed to the site of the thorn, clustering around it, engulfing it. These cells were eating the foreign object. Metchnikoff called them phagocytes, from the Greek phagon, to eat, and ketos, cell, eating cells. And he proposed that this was the fundamental mechanism of immunity, that your body defends itself by sending specialized cells to physically devour invaders. Think about what a radical idea that was. At the time, most scientists thought of individual cells as simple building blocks, like bricks in a wall. The idea that cells could act independently, could sense a threat, migrate toward it, and destroy it, that was giving cells a kind of agency, almost an intelligence, that made a lot of people uncomfortable. Metchnikoff was ridiculed by some of the most powerful figures in German science, which was the dominant scientific culture of the era. They thought his phagocyte theory was naive, overly simple, maybe even a little mystical. And the reason they were skeptical was that they had their own theory. and it was backed by some genuinely impressive evidence. This was the humoralist camp. Not humoral in the ancient Greek sense of bodily humors, but humoral meaning related to substances dissolved in the blood and body fluids. The key figures here were people like Emil von Behring and Paul Ehrlich, working in the powerhouse German laboratories of the 1890s. Von Behring, along with Kitasato Shibasaburo, had made a stunning discovery in 1890. If you took serum, the liquid part of blood with the cells removed, from an animal that had recovered from diphtheria and injected that serum into another animal, the second animal became protected against diphtheria too. No cells were transferred, just the fluid, which meant there was something dissolved in the blood, some substance, that could confer immunity all by itself. Von Behring called this substance antitoxin. We'd later call these molecules antibodies. And this discovery was so immediately, dramatically useful, antitoxin therapy saved children dying of diphtheria, that von Behring won the very first Nobel Prize in Physiology or Medicine in 1901. So you can see why the German establishment was confident. They had a therapy that worked, a Nobel Prize to show for it, and a clear mechanism. Immunity was about soluble factors in the blood, not about Metchnikoff's little eating cells. But Metchnikoff kept working, kept publishing, kept arguing. He joined the Pasteur Institute in Paris, which gave him a base outside the reach of the German establishment, and he spent decades refining his theory of cellular immunity. And here's what's really lovely about how this resolved. In 1908, the Nobel Committee essentially threw up its hands and gave the prize to both of them, Metchnikoff and Ehrlich, jointly for their work on immunity, which was the committee's way of saying, you're both right. The immune system uses both strategies. It has cellular components, cells that physically attack and engulf invaders, and it has humoral components, antibodies and other molecules dissolved in the blood that neutralize threats chemically. The full picture required both halves, and that's actually a beautiful example of something that happens a lot in the history of understanding the body. Two camps argue bitterly for decades, each with strong evidence, each convinced the other is missing the point, and the eventual answer turns out to be, the body is doing both things simultaneously, because the body is almost always more complicated than any single theory can capture. Now, let's talk about what antibodies actually are, because they're one of the most elegant pieces of molecular engineering in your entire body. An antibody is a Y-shaped protein. Picture the letter Y. The two upper arms of the Y are the business end. They're the parts that bind to a specific target, which we call an antigen. An antigen can be a protein on the surface of a bacterium, a fragment of a virus's coat, a toxin molecule, basically anything foreign that the immune system decides to target. And the key word here is specific. Each antibody binds to one particular antigen shape and one shape only. It's like a lock and key. The antibody for measles won't bind to the influenza virus. The antibody for tetanus toxin won't recognize a streptococcus bacterium. Each one is custom fitted. And here's where it gets wild. Your body can produce antibodies against essentially any molecular shape it might ever encounter. Not just the diseases that exist today, but diseases that haven't evolved yet. Synthetic chemicals that have never existed in nature. Molecules designed in a laboratory that no living thing has ever been exposed to. Your immune system can make an antibody for it. How? How can your body prepare for threats it's never seen and can't possibly predict? This was one of the great mysteries of 20th century biology, and the answer, when it finally came, was so strange and so counterintuitive that it took years for the scientific community to fully accept it. The person who figured it out was Susumu Tonegawa, a Japanese molecular biologist working at the Basel Institute for Immunology in Switzerland in the 1970s. What Tonegawa discovered was that the genes encoding antibodies don't just sit there in your DNA as fixed, complete blueprints. Instead, your immune cells do something that almost no other cells in your body do. They deliberately rearrange their own DNA. Here's how it works. In the cells that will become antibody-producing B cells, they're called B cells because they mature in the bone marrow. There are multiple gene segments that code for different parts of the antibody molecule. These segments come in families with dozens or even hundreds of slightly different versions of each segment. As a B cell matures, it randomly selects one version from each family and physically splices them together, cutting out the DNA in between and joining the chosen segments into a single functional gene. This process is called VDJ recombination, and it's essentially a controlled, deliberate mutation of the cell's own genome. Think about what that means. Each B cell assembles its antibody gene from a random combination of parts, like pulling different colored tiles from several bags and arranging them into a unique mosaic. The number of possible combinations is astronomical. Your body can generate somewhere on the order of 10 billion or more distinct antibody shapes from a finite set of gene segments. And on top of that, there's an additional process called somatic hypermutation, where the assembled gene undergoes rapid targeted mutation to fine-tune the antibody's fit to its target. It's evolution happening inside your body in real time, on a timescale of days rather than millennia. Tonegawa won the Nobel Prize in 1987 for this discovery, and it fundamentally changed how biologists thought about genetics. Before this, the dogma was that every cell in your body has the same DNA. And that's mostly true. Your liver cells and your skin cells and your neurons all carry the same genome you started with. But your B cells are the exception. Each one has deliberately altered its own DNA to create a unique antibody. You are, in a very real sense, not a single genetic entity. Your immune system is a mosaic of billions of slightly different genomes, each one a variation on your original blueprint, each one encoding a different antibody, each one a ticket in a vast lottery that might or might not match a future invader. And this brings us back to that fundamental question: how does the body know self from non-self? Because if your immune system can generate antibodies against any molecular shape, it will inevitably generate some that match your own tissues, antibodies that could attack your own cells. And this is, in fact, exactly what happens during development. Your immune system produces a huge, random repertoire of B cells, and many of them are self-reactive. They recognize your own proteins. So there has to be a quality control step, a selection process that weeds out the dangerous ones before they can cause harm. This process happens in several places, but one of the most important is the thymus, that small, unassuming gland sitting behind your breastbone that most people have never thought about. The thymus is where T cells, the other major branch of your adaptive immune system, go to be educated. And educated is really the right word, because what happens in the thymus is something like a brutal final exam. Young T cells arrive from the bone marrow, and they're presented with samples of your body's own proteins. If a T cell reacts strongly to self-proteins, it's killed, eliminated, deleted from the repertoire. Only T cells that ignore self but can still recognize foreign antigens are allowed to survive and graduate into the bloodstream. This process is called clonal deletion or negative selection, and it was worked out through decades of painstaking research by immunologists like Sir Frank McFarlane Burnett, who proposed the clonal selection theory in the late 1950s. Burnett's insight was that immunity isn't about the body learning to recognize invaders. It's about the body generating every possible recognition tool randomly, then deleting the ones that would attack itself, and keeping the rest on standby. It's a subtractive process. You don't learn what's foreign. You learn what's self, and everything else is, by default, a potential target. And when this system fails, when self-reactive cells escape the thymus or the bone marrow without being eliminated, that's autoimmune disease. That's your immune system turning its extraordinary destructive power against your own tissues. Type 1 diabetes, where immune cells destroy the insulin-producing beta cells in the pancreas. Rheumatoid arthritis, where they attack the lining of your joints. Multiple sclerosis, where they strip the insulating myelin from your nerve fibers. These are all failures of self-tolerance, breakdowns in that quality control process, and they affect tens of millions of people worldwide. But there's another side to immune recognition that's equally fascinating, and it connects to something much older than the adaptive immune system we've been talking about. Because antibodies and T cells and all of that sophisticated, customizable machinery, that's a relatively recent evolutionary invention, found only in vertebrates. For most of life's history, organisms defended themselves with a different, more ancient system, one that doesn't rely on random gene rearrangement or clonal selection at all. It's called innate immunity, and it works on a completely different principle. Instead of generating random receptors and then selecting the useful ones, innate immune cells come pre-equipped with a fixed set of receptors that recognize common molecular patterns shared by broad categories of pathogens. Patterns like the lipopolysaccharide found in the outer membrane of many bacteria, or the double-stranded RNA that certain viruses produce during replication. These aren't specific to any one pathogen. They're general danger signals, molecular fingerprints of entire classes of invaders, and the person who really cracked this open, who identified the specific receptors that innate immune cells use to detect these patterns, was Jules Hoffmann, working with fruit flies in Strasbourg in the 1990s. Hoffmann discovered that a gene called Toll, which had originally been identified as important for fruit fly development, was actually critical for the fly's immune defense against fungal infections. Flies with mutant Toll genes couldn't fight off fungi and died. And then Bruce Beutler, working independently in Texas, found the mammalian equivalent, Toll-like receptors, or TLRs, and showed that they played the same role in mice and humans. These Toll-like receptors are your first line of defense, the sentinels that detect an invasion within minutes and trigger the initial inflammatory response, while your adaptive immune system, with its antibodies and T cells, takes days to ramp up its targeted response. So innate immunity is fast but blunt, and adaptive immunity is slow but precise, and your body needs both of them working together, handing off information from one system to the other, like relay runners passing a baton. And that handoff, that's where things get really elegant. Because the innate immune system doesn't just fight pathogens on its own. It also tells the adaptive immune system what to fight and how urgently. The cells that do this most effectively are called dendritic cells, and they were discovered by a man named Ralph Steinman at Rockefeller University, starting in the early 1970s. Steinman noticed these strange, spindly cells with long branching arms. That's where the name comes from. They look a bit like the dendrites on nerve cells, and he spent decades arguing that they were the critical link between innate and adaptive immunity. For years, most immunologists didn't take him seriously. They thought dendritic cells were just some minor cell type, not worth much attention. But Steinman kept at it, kept publishing, kept refining his case, and eventually the evidence became overwhelming. Dendritic cells sit in your tissues, your skin, your gut lining, your lungs, and they're constantly sampling the environment, picking up bits of whatever's around them. When a toll-like receptor on a dendritic cell detects a pathogen, the cell swallows it up, chops it into fragments, and then migrates to the nearest lymph node, where it presents those fragments on its surface to T cells. It's essentially showing the adaptive immune system a mugshot of the invader and saying, "This is what you need to go after, and here's how dangerous it is." Without that presentation step, your T cells wouldn't know what to attack. Steinman was awarded the Nobel Prize in Physiology or Medicine in 2011 for this work. But here's the bittersweet part. He died of pancreatic cancer just 3 days before the Nobel Committee made their announcement. They didn't know he had passed when they made the decision. The Nobel Prize is almost never awarded posthumously, but the committee decided to honor it anyway because the decision had been made in good faith while they believed he was alive. Steinman had actually used his own discoveries to help design experimental immunotherapy treatments for his cancer, and his colleagues believe those treatments extended his life by several years beyond what anyone expected. So in a very real sense, his understanding of how the immune system communicates bought him time, not enough, but more than he would have had otherwise. Now, this whole conversation about immunity, innate and adaptive, antibodies and T cells, dendritic cells bridging the gap, it sets up one of the most important practical questions in the history of medicine. How do you train the immune system to fight something it hasn't encountered yet? And that brings us back to vaccination, which we touched on earlier with Jenner and his cowpox experiment in 1796. But the story of vaccination after Jenner is its own sprawling saga, and it's worth spending a little time with because it shows how understanding the body's mechanisms leads directly to engineering solutions that save millions of lives. Jenner knew his method worked, but he didn't really know why. He couldn't have. The germ theory of disease wasn't established yet, and the concept of an immune system as we understand it was more than a century away. The next great leap came from Louis Pasteur in the 1880s. Pasteur was working with chicken cholera, a disease caused by bacteria, and he stumbled onto something remarkable, almost by accident. He had some old bacterial cultures that had been sitting around in his lab over the summer, weakened by exposure to air. When he injected chickens with these aged, weakened bacteria, the chickens got mildly sick, but recovered. And then, here's the key. When he later injected those same chickens with fresh, virulent bacteria, they didn't get sick at all. They were protected. Pasteur realized that by weakening the pathogen, you could teach the body to recognize and fight it without causing the full disease. He called this process vaccination, in honor of Jenner's earlier work with vaccinia, the cowpox virus. And he went on to develop vaccines for anthrax and rabies using similar principles of attenuation, taking a dangerous pathogen and weakening it just enough that it could provoke an immune response without causing serious harm. What Pasteur was doing, even though he didn't have the vocabulary for it yet, was exploiting exactly the mechanism we've been talking about. He was giving the adaptive immune system a preview. The weakened pathogen carried the same molecular shapes, the same antigens, as the dangerous version, so when dendritic cells picked it up and presented it to T cells and B cells, the body mounted a real immune response, generated memory cells, and was ready for the actual threat whenever it showed up. That's all a vaccine is, really. It's a rehearsal. You're letting your immune system practice on a safe version of the fight so it's ready for the real one. And that principle, give the body a harmless preview of a dangerous pathogen, has been the foundation of vaccinology for over a century. But the engineering challenge is enormous. How do you weaken a pathogen enough to be safe, but keep it similar enough to be useful? How do you make sure it doesn't revert to a dangerous form? How do you preserve it, transport it, scale production to millions of doses? These are deeply practical problems, and solving them has required as much ingenuity as any bridge or aircraft. Take the polio vaccine, one of the most consequential medical engineering projects of the 20th century. In the early 1950s, polio was terrifying. It paralyzed tens of thousands of children every year in the United States alone, and parents lived in dread every summer when outbreaks peaked. Two researchers took fundamentally different approaches to the problem, and their rivalry became one of the most famous in medical history. Jonas Salk, working at the University of Pittsburgh, developed a vaccine using poliovirus that had been killed, completely inactivated with formaldehyde. It couldn't replicate at all, so it was very safe, but it still carried the right antigens to provoke an immune response. Albert Sabin, on the other hand, developed an oral vaccine using live poliovirus that had been attenuated, weakened by passage through non-human cells until it could no longer cause disease in people. Sabin's vaccine had certain advantages. It was easier to administer, just drops on the tongue, no needle. And because it used a live virus that replicated briefly in the gut, it produced stronger mucosal immunity and could even spread passively to unvaccinated people nearby, creating a kind of secondary immunization effect. But it carried a tiny risk. In very rare cases, about 1 in every 2.4 million doses, the weakened virus could mutate back toward virulence and actually cause the disease it was supposed to prevent. Salk's killed vaccine went first. The 1954 field trial was one of the largest medical experiments in history. Nearly 2 million children participated, and the logistics alone were staggering. When the results were announced, on April 12, 1955, confirming the vaccine was safe and effective, church bells rang across America. Salk became a national hero overnight. But then, just weeks later, disaster. A manufacturing defect at Cutter Laboratories in Berkeley, California, resulted in batches of vaccine that contained live, active poliovirus. The inactivation process hadn't been completed properly. About 120,000 children received these defective doses. 40,000 developed mild polio. 164 were severely paralyzed. 10 died. It was called the Cutter incident, and it was devastating, not just for the families affected, but for public trust in vaccination itself. The episode led to a complete overhaul of vaccine manufacturing oversight in the United States and the creation of much stricter regulatory protocols. It's a painful reminder that in biological engineering, quality control isn't just a nice-to-have. It's the difference between saving lives and taking them. Sabin's oral vaccine eventually became the dominant form used worldwide, particularly in mass immunization campaigns in developing countries because of its ease of administration. But as polio was driven closer and closer to eradication, that tiny risk of vaccine-derived poliovirus became a bigger concern proportionally. In countries where wild polio had been eliminated, the only cases of polio were being caused by the vaccine itself. So many countries, including the United States in 2000, switched back to Salk's inactivated vaccine, which carries no risk of reversion. The engineering trade-offs shifted as the epidemiological landscape changed. What was the right choice in 1960 wasn't necessarily the right choice in 2000. That's something you see across all kinds of engineering. The optimal solution depends on the context, and the context is always changing. But here's what's really wild about where vaccine science has gone since then. For most of the 20th century, vaccines were made from whole pathogens, either killed or weakened. You were working with the actual organism. But starting in the 1980s and accelerating dramatically in the 2000s and 2010s, researchers began developing entirely new platforms. Instead of growing a virus, weakening it, and injecting it, what if you could just give the body the genetic instructions to make one specific protein from the virus, just enough for the immune system to recognize and remember without any of the other viral machinery? That's the idea behind mRNA vaccines, and the story of how they came to be is one of the most remarkable in modern biomedical engineering. The key figure here is Katalin Karikó, a Hungarian-born biochemist who spent decades working on mRNA at the University of Pennsylvania, often in near obscurity. Karikó believed that synthetic messenger RNA could be used therapeutically, that you could design an mRNA molecule encoding a specific protein, deliver it into cells, and let the cell's own ribosomes read the instructions and build that protein. The concept was elegant, but there was a massive problem. When you inject synthetic mRNA into the body, the innate immune system, those toll-like receptors we were just talking about, recognizes it as foreign and mounts an inflammatory response that destroys the mRNA before it can do its job. The very defense system that protects you from viral RNA was treating the therapeutic mRNA as a threat. Karikó and her collaborator Drew Weissman discovered something crucial in 2005. They found that by modifying one of the nucleoside bases in the synthetic mRNA, swapping uridine for pseudouridine, they could slip the mRNA past the innate immune sentinels without triggering that inflammatory alarm. The modified mRNA was still perfectly readable by ribosomes, still produced the desired protein, but it was essentially invisible to toll-like receptors. It was a biochemical disguise, and it solved the fundamental problem that had stalled the entire field. For years, almost nobody paid attention. Karikó had her grant applications rejected repeatedly. She was demoted from her faculty position. She kept going anyway, convinced the science was sound. And then, in early 2020, when the genetic sequence of a novel coronavirus was published by Chinese researchers in January, companies like BioNTech and Moderna were able to design mRNA vaccines encoding the virus's spike protein within days, literally days, using the platform that Karikó and Weissman had. Spent decades developing. The speed was breathtaking, from sequence to candidate vaccine in under a week. Clinical trials, manufacturing scale-up, and emergency authorization within a year. It was possible because the hard foundational work had already been done, quietly, by people the scientific establishment had largely overlooked. Karikó and Weissman shared the Nobel Prize in Physiology or Medicine in 2023. And the beauty of their discovery is that it connects everything we've been talking about: the innate immune system's pattern recognition, the adaptive immune system's ability to learn from a preview, the ribosomal machinery that translates mRNA into protein. The vaccine works because it speaks the body's own language. It hands your cells a set of instructions written in the same molecular code your cells already use, and your cells do the rest. Your ribosomes build the spike protein. Your dendritic cells pick it up and present it. Your T cells and B cells respond, and memory cells form. The whole elegant cascade we traced from Mechnikov's phagocytes to Tonegawa's gene rearrangement to Steinman's dendritic cells, all of it activates, just as it would for a real infection, but without the danger. Now, The immune system is extraordinary, but it doesn't operate in isolation. It's deeply connected to something we haven't really explored yet: the nervous system's role in sensing and responding to damage within the body. And that brings us to a topic that's surprisingly poorly understood, given how universal the experience is: pain. Everyone knows what pain feels like, but the question of how your body actually generates that sensation, how a stubbed toe or a burned finger translates into an experience you can't ignore, that's a story that took centuries to even begin to untangle. For the longest time, the dominant idea was almost absurdly simple. Pain was thought to be a direct line, like a bell pull in a Victorian house. You tug the rope at one end, the bell rings at the other. Damage happens in your finger, a signal shoots straight up to your brain, and you feel pain. One-to-one, mechanical, straightforward. And honestly, that model isn't entirely wrong. It captures something real about the system, but it misses so much. It misses why a soldier can take a bullet on a battlefield and feel nothing for hours, then collapse in agony later. It misses why a paper cut can sometimes hurt more than a deep bruise. It misses why your back can ache for years after the original injury has completely healed. The simple bell-pull model can't explain any of that. And the journey from that naive picture to our current understanding is one of the most fascinating and humbling stories in the history of physiology. Let's start with the basics, the part that actually does work like a bell pull. You have specialized nerve endings scattered throughout your body, in your skin, your muscles, your joints, your organs. And these nerve endings are called nociceptors. The name comes from the Latin word nocere, meaning to harm. They're harm detectors. And they were first clearly described by Charles Sherrington, the great British neurophysiologist, around the turn of the 20th century. Sherrington was working at the University of Liverpool in the 1890s and early 1900s, and he was obsessed with reflexes, the automatic responses your body makes before your conscious mind even gets involved. Pull your hand away from a hot stove before you've consciously registered the heat. That's a reflex, and Sherrington mapped out how these reflex arcs work with extraordinary precision. He shared the Nobel Prize in 1932 for his work on the function of neurons, and one of his key contributions was the idea that there had to be a specific class of sensory nerve fiber dedicated to detecting harmful stimuli. Not just any touch, not pressure or temperature in the normal range, but specifically things that threaten tissue damage. And he was right. Your nociceptors are tuned to respond to intense mechanical force, extreme temperatures, both hot and cold, and certain chemicals that are released when cells are damaged. Think about what happens when you stub your toe. The impact crushes some cells. Those crushed cells spill their contents, releasing molecules like potassium ions, ATP, and prostaglandins. These chemicals wash over the nociceptor nerve endings in the area, and the nociceptors respond by generating electrical signals, action potentials, that travel along nerve fibers toward your spinal cord. Now here's a detail that matters more than you might think. There are two main types of nerve fibers that carry pain signals, and they work at very different speeds. The fast ones are called A-delta fibers. They're thinly coated in myelin, that insulating sheath we talked about earlier, and they conduct signals at maybe 5 to 30 meters per second. These are the ones responsible for that sharp, immediate, localized sting you feel the instant you stub your toe. You know exactly where it hurts, and you know it right away. But then, a second or two later, a different kind of pain arrives. A slower, deeper, more diffuse ache that's harder to pinpoint. That's carried by C fibers, which are unmyelinated, no insulating sheath, and they conduct signals much more slowly, maybe 0.5 meter to 2 meters per second. So, you actually feel pain in two waves. The fast, sharp signal arrives first via A delta fibers, and the slow, burning, throbbing signal follows via C fibers. Next time you bump your elbow hard, pay attention. You'll notice it. That initial flash of sharp pain, and then a moment later, the deeper ache rolling in behind it. Two separate fiber types, two separate speeds, two distinct sensations from the same event. These signals travel along the nerve fibers into your spinal cord, entering through the dorsal horn, the back part of the spinal cord. And this is where things start to get really interesting, because the spinal cord is not just a passive relay station. It's not simply passing the signal along unchanged. The dorsal horn is a processing center, and what happens there can amplify, dampen, or fundamentally alter the pain signal before it ever reaches your brain. This is the insight that transformed our understanding of pain, and it came from a remarkable collaboration between two scientists, Ronald Melzack, a Canadian psychologist, and Patrick Wall, a British neuroscientist. In 1965, they published what's known as the gate control theory of pain, and it was genuinely revolutionary. Here's the idea. Melzack and Wall proposed that there's essentially a gate in the spinal cord, not a literal physical gate, but a functional one created by the interaction of different types of nerve signals. Pain signals coming in through those small C fibers, and A delta fibers tend to open the gate, allowing the pain message to pass through and travel up to the brain. But signals coming in through larger nerve fibers, the ones that carry ordinary touch and pressure information, the kind activated when you rub your skin or apply pressure, those signals tend to close the gate. They inhibit the transmission of pain signals. And this is why rubbing a bumped knee actually helps. You're not imagining it. When you rub the area around an injury, you're activating large diameter touch fibers, and those signals partially close the gate in the dorsal horn, reducing the pain signal that reaches your brain. It's a real physiological mechanism, not just distraction. But Melzack and Wall went further. They proposed that the gate could also be influenced from above by signals descending from the brain itself. Your emotional state, your attention, your expectations, your past experiences, all of these can send signals down from the brain to the spinal cord that modulate the gate, opening it wider or pushing it closed. This is why context matters so much for pain. That soldier on the battlefield who doesn't feel his wound, his brain is sending powerful descending inhibitory signals that are slamming that gate shut, because in that moment, survival requires focus and action, not the distraction of pain. The pain will come later, when the danger has passed and the brain's priorities shift. And conversely, anxiety and fear can open the gate wider, amplifying pain signals. If you're anxious in a dentist's chair, expecting pain, the pain you feel from a procedure can genuinely be worse, not because the stimulus is different, but because your brain is modulating the gate in a way that lets more signal through. Now, the gate control theory wasn't perfect in every detail. The specific neural circuits Melzack and Wall proposed have been refined and revised over the decades. But the core insight, that pain is not a simple bottom-up signal, but is actively modulated at multiple levels, that insight has held up beautifully, and it opened the door to understanding some of the most puzzling phenomena in medicine. Take phantom limb pain. Someone loses an arm or a leg, and yet they feel excruciating pain in the limb that's no longer there. Under the old Bell-Pull model, this made no sense at all. There's no limb, there are no nociceptors, there's no tissue damage. So where's the pain coming from? Melzack spent much of his career wrestling with this question, and he eventually proposed what he called the neuromatrix theory. The idea that pain is ultimately generated by a widely distributed network of neurons in the brain, and that this network can produce the experience of pain even in the absence of any input from the body. The brain has an internal model of the body, a kind of neural map, and that map doesn't simply disappear when a limb is amputated. The brain regions that used to process signals from that limb are still there, still active, and they can generate pain signals on their own, or they can be triggered by reorganized inputs from neighboring body regions. It's the brain itself creating the pain, not the body. And this connects to something even more common and clinically important: chronic pain. Millions of people live with pain that persists long after the original injury is healed. The tissue damage is gone, the inflammation has resolved, but the pain continues, sometimes for years, sometimes for a lifetime. For a long time, medicine struggled with this. If there's no ongoing damage, what's causing the pain? The answer, we now understand, involves a phenomenon called central sensitization. When pain signals are transmitted repeatedly or intensely, the neurons in the dorsal horn of the spinal cord can undergo lasting changes. Their threshold for firing drops. They become more excitable. Signals that would normally be too weak to trigger a pain response start getting through. Even ordinary touch, a light brush against the skin, can be interpreted as painful. This is called allodynia, and if you've ever had a bad sunburn where even the touch of a shirt against your skin was agonizing, you've experienced a temporary version of it. In chronic pain conditions, this sensitization can become semi-permanent. The nervous system essentially learns to be in pain. And here's what's really sobering about that. It means the pain is completely real. It's not imagined. It's not psychological in the dismissive sense that word is sometimes used. But it's being generated by a nervous system that has changed its own wiring. The alarm system has malfunctioned. It's firing when there's no fire. And treating it requires understanding that you're not treating an injury anymore. You're treating a nervous system that has been reprogrammed. This is where modern pain science gets both fascinating and deeply challenging. Because the molecules involved in pain signaling are themselves incredibly complex. One of the most important discoveries came in 1997 when David Julius at the University of California, San Francisco, identified the receptor that makes chili peppers burn. It's called TRPV1, transient receptor potential vanilloid 1, and it's a protein sitting on the surface of nociceptor nerve endings. Capsaicin, the chemical that makes chili peppers hot, binds to this receptor and activates it, which is why eating a hot pepper feels like burning even though no actual thermal damage is occurring. But here's the beautiful part. TRPV1 isn't there to detect chili peppers. It's a heat sensor. It activates at temperatures above about 43 degrees Celsius, the threshold where heat starts to become damaging. Capsaicin just happens to fit into the same receptor. Julius went on to identify another receptor, TRPM8, that detects cold, and that's the receptor activated by menthol, which is why mint feels cool on your tongue even at room temperature. Julius shared the Nobel Prize in 2021 with Ardem Pataputian, who discovered the piezo receptors, the mechanosensors that detect pressure and touch. Piezo 1 and piezo 2 are proteins that sit in cell membranes and physically change shape when the membrane is stretched or pressed, opening an ion channel that generates an electrical signal. These are the molecules that let you feel the weight of a blanket on your skin, the pressure of your feet against the floor, the stretch of a full stomach. So between Julius's temperature and chemical receptors and Pataputian's mechanical sensors, we now have a molecular-level picture of how the body's sensory nerve endings convert physical stimuli into electrical signals. It's transduction, the same basic principle we talked about with the kidneys sensing blood pressure or the endocrine system detecting hormone levels. The body is constantly converting one kind of signal into another, and at the sensory periphery, these receptor proteins are the translators. But pain doesn't end at detection. Once those signals reach the brain, traveling up through the spinal cord, through the brainstem, through the thalamus, they fan out into a whole constellation of brain regions. There's no single pain center in the brain. The somatosensory cortex processes the location and intensity. The anterior cingulate cortex and the insula contribute the emotional dimension, the unpleasantness, the suffering. The prefrontal cortex adds context and meaning. And the descending pathways we talked about with Melzack and Wall loop back down, modulating the incoming signals. The brain's own opioid system, endorphins and enkephalins, can powerfully suppress pain through these descending pathways. That runner's high, that strange calm after an injury, that's your endogenous opioid system at work, and it acts on many of the same receptors that morphine does, which, of course, is why opioid drugs are so effective at killing pain and so devastatingly addictive. And this brings up something worth sitting with for a moment. Pain is protective. It's one of the most important survival mechanisms you have. There are rare individuals born with a condition called congenital insensitivity to pain, caused by mutations in genes like SCN9A that encode sodium channels critical for nociceptor function. These people can't feel pain at all, and far from being lucky, they're in constant danger. They break bones without noticing. They burn themselves severely. They develop joint damage from injuries they never felt. Many don't survive to adulthood. Pain exists because organisms that could feel it survived, and organisms that couldn't were destroyed by damage they never detected. It's an ancient, essential warning system. The problem is that the system can malfunction, can become chronic, can outlast its usefulness, can itself become a source of suffering rather than protection. And understanding that distinction between acute protective pain and chronic maladaptive pain is one of the great ongoing challenges of modern medicine. Now, pain is just one example of something broader, your body's constant internal monitoring system, the way it keeps track of its own state. And this connects to a concept that's been quietly running underneath everything we've talked about tonight, from breathing to blood pressure to hormone levels to immune responses. It's the idea of homeostasis, the body's relentless drive to maintain stable internal conditions. And the person who gave us that word, who crystallized that concept into something the whole field could build on, was a physiologist named Walter Bradford Cannon, working at Harvard in the early 20th century. Cannon was one of those figures who just kept showing up at the edges of nearly every topic we've touched on tonight. He studied the autonomic nervous system. He coined the phrase fight or flight, and in 1932, he published a book called The Wisdom of the Body, where he laid out this idea that your internal environment isn't just passively sitting there, it's being actively, continuously regulated. He called that regulation homeostasis, from the Greek words for same and standing. And the name stuck because it captured something profound. Your body doesn't just happen to be at 98.6 degrees Fahrenheit. It fights to stay there. It doesn't just happen to have a blood pH of about 7.4. It defends that number with everything it's got. And the reason Cannon's idea was so powerful is that it reframed the body not as a collection of parts doing their own thing, but as an integrated system where everything is connected to everything else through feedback loops, loops that sense a change, respond to it, and then check whether the response worked. Now, Cannon was building on earlier work. The French physiologist Claude Bernard, back in the 1850s and 1860s, had already articulated the idea that the body maintains a stable internal environment, what he called the milieu intérieur, the internal milieu. Bernard's insight was revolutionary for its time. He recognized that your cells don't interact directly with the outside world. They live in a carefully controlled internal sea. Your blood, your interstitial fluid, the fluid between cells, and the composition of that sea matters enormously. If it changes too much, cells die. So Bernard said, in what became one of the most quoted lines in all of physiology, "The constancy of the internal environment is the condition for free and independent life." What he meant was that the reason you can walk around in the desert, or swim in cold water, or skip a meal, or run a marathon, is that your body keeps its internal conditions stable regardless of what's happening outside. You're not at the mercy of the environment the way a single-celled organism floating in a pond is. You carry your own environment with you, inside you, and you regulate it. Cannon took that idea and gave it teeth. He described the specific mechanisms, the feedback loops, the sensors, the effectors. And here's the thing. Almost everything we've talked about tonight is a homeostatic system. When we talked about breathing, about how your brainstem monitors carbon dioxide levels and adjusts your breathing rate, that's homeostasis. When we talked about your kidneys adjusting how much water and sodium they retain to keep your blood pressure and electrolyte balance steady, homeostasis. When we talked about insulin and glucagon managing your blood sugar, homeostasis. Your body is running hundreds of these feedback loops simultaneously, every second of every day, and you're not aware of almost any of them. So let's talk about how these loops actually work, because the engineering is elegant. Most homeostatic systems use what's called negative feedback. And that word negative is a little misleading. It doesn't mean bad. It means the system responds to a change by pushing back in the opposite direction. Think of a thermostat in your house. You set it to 70 degrees. The temperature drops to 68. The thermostat senses the deviation, turns on the heater, the temperature rises back to 70, and the heater shuts off. If the temperature climbs to 72, the system kicks on the air conditioning, brings it back down. The response is always opposite to the disturbance. That's negative feedback. And your body uses this principle everywhere. Temperature regulation is actually a perfect example, and it's more sophisticated than any thermostat you'll find in a building. Your hypothalamus, that small region at the base of your brain that we mentioned when we talked about hormones, acts as your body's thermostat. It has neurons that are directly sensitive to the temperature of the blood flowing through it, and it also receives signals from temperature receptors in your skin, your spinal cord, your abdominal organs. It integrates all of that information and compares it to a set point, which for most people is right around 37 degrees Celsius, or 98.6 Fahrenheit, though there's actually more individual variation than people realize, and your set point shifts throughout the day. It's lower in the morning and higher in the late afternoon. When your core temperature starts to rise, say you're exercising or you're out in the heat, the hypothalamus triggers a cascade of cooling responses. Blood vessels near the surface of your skin dilate, which is called vasodilation, and that brings warm blood closer to the surface where heat can radiate away. You start sweating, and as that sweat evaporates, it pulls heat off your skin. Evaporative cooling. Same principle that makes you feel cold when you step out of a swimming pool on a breezy day. Your behavior changes too. You feel hot, you seek shade, you want cold water. All of that is orchestrated by the hypothalamus. When your core temperature drops, the opposite happens. Skin blood vessels constrict, keeping warm blood deeper in your body, away from the cold surface. You start shivering. Those rapid, involuntary muscle contractions generate heat as a byproduct, because muscle contraction is only about 25% mechanically efficient, and the other 75% of the energy becomes heat. You get goosebumps, which in our fur-covered ancestors would have fluffed up the fur to trap an insulating layer of air. Doesn't do much for us anymore. But the reflex is still there. And again, your behavior shifts. You curl up, you put on a blanket, you huddle. Here's what's really clever about this system. It's not just reactive, it's anticipatory. Your body doesn't always wait for the temperature to change before responding. If you eat a large meal, your metabolic rate is about to increase, which will generate heat. Your body starts adjusting before the temperature actually rises. If you step outside on a cold day, temperature receptors in your skin detect the cold air before your core temperature has dropped at all, and they start triggering conserving responses preemptively. This is sometimes called feed-forward control, and it's layered on top of the negative feedback system to make the whole thing faster and more precise. And fever. Fever is fascinating because it's not a failure of temperature regulation. It's a deliberate resetting of the set point. When your immune system detects an infection, immune cells release signaling molecules called pyrogens, interleukin-1, interleukin-6, tumor necrosis factor, and these act on the hypothalamus and essentially tell it, raise the set point. Now instead of defending 37 degrees, your hypothalamus is defending 39 degrees. So even though your body temperature is normal, the hypothalamus thinks it's too cold. You feel chilled, you shiver, you pile on blankets. Your body generates and conserves heat until it reaches the new, higher set point. And the reason your body does this is that many pathogens replicate more slowly at higher temperatures, and many immune processes work more efficiently when you're warmer. Fever is a weapon. It's your immune system commandeering your thermostat for tactical advantage, which is why, in many cases, moderate fevers don't necessarily need to be suppressed. They're doing something useful. Though obviously, extremely high fevers are dangerous and need treatment. Now, temperature is one variable, but think about all the others. Blood glucose, blood pressure, blood oxygen, blood pH, calcium levels, sodium levels, potassium levels, water balance. Each one has its own sensors, its own control center, its own set of effectors, and its own set point. And they're not independent. They interact. If you're dehydrated, that affects your blood pressure, which affects your kidney function, which affects your electrolyte balance, which affects your nerve and muscle function. Pull on one thread and you tug on a dozen others. The body has to coordinate all of these simultaneously, and it does it through the interplay of the nervous system and the endocrine system that we talked about earlier. Fast signals through nerves for immediate adjustments, slower signals through hormones for sustained regulation. And here's something worth appreciating. The precision of this regulation is staggering. Your blood pH is maintained between about 7.35 and 7.45. That's an incredibly narrow range. If it drops below 7.0 or rises above 7.8, you can die. Your body uses 3 overlapping systems to keep it in range: chemical buffers in the blood that absorb excess acid or base almost instantly, respiratory adjustments where you breathe faster to blow off carbon dioxide, which is acidic when dissolved in blood, or slower to retain it, and renal adjustments where your kidneys excrete or retain hydrogen ions and bicarbonate over hours to days. 3 systems, 3 different timescales, all defending the same variable. That's redundancy. That's robust engineering. Or think about calcium. You need calcium for muscle contraction, nerve signaling, blood clotting, bone structure. It's essential. And your blood calcium level is regulated with extraordinary precision by parathyroid hormone and calcitonin, working in opposition. Parathyroid hormone raises blood calcium by pulling it from bone, increasing absorption from food, and reducing how much your kidneys excrete. Calcitonin does roughly the opposite. And the parathyroid glands, 4 tiny glands embedded in the back of your thyroid, are directly sensing blood calcium levels and adjusting their hormone output minute by minute. This is so precisely controlled that surgeons operating on the thyroid have to be incredibly careful not to damage the parathyroids, because if you lose them, your calcium regulation collapses and the consequences are severe. Muscle spasms, cardiac arrhythmias, seizures. There's a concept in engineering called robustness, the ability of a system to continue functioning despite perturbations, despite things going wrong. And your body is remarkably robust, partly because of redundancy, partly because of the layered feedback systems, and partly because of something called allostasis. This is a term introduced by the neuroscientist Peter Sterling and the epidemiologist Joseph Ayer in 1988, and it extends Cannon's homeostasis in an important way, where homeostasis emphasizes stability, keeping things the same. Allostasis emphasizes stability through change. The idea is that your body doesn't just maintain fixed set points. It actively predicts what you're going to need and adjusts in advance. Before you wake up in the morning, your cortisol levels start rising to prepare you for the metabolic demands of the day. Before you start exercising, your heart rate begins to increase. Your body is constantly modeling the future and pre-adjusting. Sterling argued that this is more accurate than the simple thermostat model. Your body isn't just reacting to deviations, it's anticipating them. But allostasis has a cost. When the system is constantly being pushed by chronic stress, by sleep deprivation, by poor nutrition, by sustained psychological pressure, the wear and tear accumulates. Sterling and later Bruce McEwen at Rockefeller University called this allostatic load. And high allostatic load is associated with a whole cascade of health problems: cardiovascular disease, immune suppression, metabolic syndrome, depression. Your regulatory systems are still working, but they're working so hard for so long that the machinery itself starts to degrade. It's like running an engine at redline for months. It'll keep running, but parts will wear out faster than they should. And this connects to something that's been quietly present in almost every system we've explored tonight. The idea that your body isn't a machine with fixed parts. It's adaptive. It remodels itself. Your bones remodel in response to the loads placed on them. This is Wolff's law, described by the German anatomist Julius Wolff in 1892. Astronauts lose bone density in microgravity because without gravitational loading, the body decides it doesn't need as much bone and starts breaking it down. Your muscles grow when you challenge them and atrophy when you don't. Your brain rewires its neural connections based on experience. Neuroplasticity, which was once considered impossible in adults, but is now understood to continue throughout life, though it slows with age. Even your immune system, as we talked about, learns and remembers. This adaptability is one of the most remarkable features of biological systems, and it's something that engineers have spent decades trying to replicate in artificial systems. Self-healing materials, adaptive algorithms, machine learning. These are all, in a sense, attempts to capture what your body does naturally. But there's a flip side to adaptability, and it's one that matters just as much as the adaptability itself. Your body can adapt in directions that aren't helpful. That's the quiet tragedy of chronic pain, actually. Remember how we talked about central sensitization? The way your nervous system can learn to amplify pain signals until harmless touch registers as agony? That's adaptability. That's your nervous system doing exactly what it's designed to do. Noticing a pattern, reinforcing it, getting better at it. It's just getting better at something you desperately wish it would stop doing. The same plasticity that lets a musician's brain reorganize to give more cortical real estate to their fingertips can, in different circumstances, reorganize to maintain pain that no longer serves any protective purpose. The system doesn't distinguish between useful adaptation and harmful adaptation. It just adapts. And you see this pattern everywhere once you start looking. Your immune system, that brilliant, layered defense we spent so much time with earlier, the one with Mechnikov's phagocytes and Tonagawa's recombining antibodies and Steinman's dendritic cells orchestrating the whole response, that same system can turn on your own tissues. Autoimmune diseases are, in a very real sense, your immune system being too good at its job, or rather, good at its job, but aimed at the wrong target. In rheumatoid arthritis, your immune cells attack the lining of your joints. In type 1 diabetes, they destroy the insulin-producing beta cells of your pancreas. In multiple sclerosis, they go after the myelin sheath that insulates your nerve fibers, that same myelin that makes nerve signals fast and efficient. The system that's supposed to protect you becomes the thing that harms you, and it does so with all the sophisticated machinery of adaptive immunity, specific, targeted, and relentless. For a long time, the question of why autoimmunity happens was genuinely mysterious. If your immune system goes through all that trouble during development to eliminate self-reactive cells, the process of clonal deletion that Burnett theorized and that subsequent research confirmed, then how do autoimmune diseases exist at all? The answer, it turns out, is that the screening process isn't perfect. Some self-reactive immune cells slip through. In healthy people, there are additional layers of control. Regulatory T cells, for instance, which act as a kind of immune police force, suppressing other immune cells that start attacking self-tissues. These regulatory T cells were controversial for years. The idea that there were dedicated suppressor cells in the immune system was proposed, then largely discredited in the 1980s, then revived in the mid-1990s when Shimon Sakaguchi in Japan identified a specific population of T cells marked by a molecule called CD25 that genuinely did suppress immune responses. It was a beautiful vindication. The concept had been right. The earlier evidence had just been muddled. So your immune system has both an accelerator and a brake. Autoimmunity often happens when the brake fails, or when some environmental trigger, an infection, a toxin, sometimes even a period of extreme stress, kicks the accelerator hard enough that the brakes can't keep up. There's a concept called molecular mimicry, where a pathogen happens to have surface proteins that look similar to your own tissue proteins. Your immune system mounts a response against the pathogen, and some of those activated immune cells cross-react with your own tissues. The infection clears, But the autoimmune response continues. Your body's own adaptability, its own exquisite specificity, becomes the problem. Here's the thing, though, and this is something worth sitting with for a moment. These failure modes aren't really failures of design. They're the inevitable trade-offs of a system that has to be both powerful and flexible. An immune system that never attacked self-tissue would probably also be too cautious to fight novel pathogens effectively. A nervous system that couldn't amplify signals would miss subtle threats. A stress response that never activated would leave you unable to respond to genuine emergencies. The problems arise at the edges, in the extreme cases, when systems that evolved for one set of conditions are pushed into situations they weren't optimized for. And that word, evolved, matters. Because your body wasn't engineered in the way we usually think about engineering. There was no designer sitting down with specifications and tolerances and safety margins. What you have is the product of billions of years of natural selection, which is an optimization process, but a very particular kind. It optimizes for reproductive success in ancestral environments, not for comfort, not for longevity, not for happiness, and certainly not for the modern world. This mismatch between what your body evolved for and how you actually live is one of the most important ideas in modern medicine. It's sometimes called evolutionary mismatch, or the mismatch hypothesis, and it helps explain a surprising number of conditions that plague modern humans. Think about your stress response. Cannon's fight-or-flight system that we talked about, the sympathetic nervous system flooding your body with adrenaline and cortisol. That system evolved for acute physical threats, a predator, a rival, a natural disaster. Short, intense bursts of danger followed by resolution. But in modern life, the threats are chronic and psychological. Financial pressure, work deadlines, social conflict, the low-grade hum of anxiety that comes from a 24-hour news cycle. Your hypothalamic pituitary adrenal axis doesn't know the difference. It responds to a looming mortgage payment with the same cascade of cortisol that it would use for a charging animal. And because the threat never resolves, because you can't fight or flee from an inbox full of emails, the system stays activated. That's McEwen's allostatic load in action. Your body is faithfully executing its programming. The programming just wasn't written for this world. Or think about diet. Your body is exquisitely good at storing energy as fat. This was a tremendous advantage for most of human history when food was scarce and unpredictable. The ability to efficiently convert surplus calories into fat reserves and then mobilize those reserves during lean times was literally the difference between life and death. Your insulin system, your adipose tissue, your leptin and ghrelin signaling, all of it is optimized for an environment where the problem was getting enough food, not having too much. Now, in many parts of the world, the problem has inverted. Calorie-dense food is cheap and abundant, and your body, still running the ancient software, keeps storing, keeps craving sugar and fat, keeps treating every meal as though the next famine might be around the corner. The obesity epidemic isn't a failure of willpower. It's a mismatch between a Stone Age metabolism and a modern food environment. Your body is doing exactly what it evolved to do. The context has just changed dramatically. Even your sleep, the very thing you might be drifting toward right now, is shaped by this evolutionary history. Your circadian rhythm, that internal clock governed by the suprachiasmatic nucleus in your hypothalamus, evolved to synchronize with natural light-dark cycles. Sunlight in the morning suppresses melatonin and promotes wakefulness. Darkness in the evening triggers melatonin release and prepares you for sleep. But we've flooded our evenings with artificial light, especially the blue spectrum light from screens, which is precisely the wavelength that most effectively suppresses melatonin. Your ancient clock is getting conflicting signals, and the result is the epidemic of sleep disruption that characterizes modern life. The system works beautifully. It's just receiving inputs it was never designed to handle. And this brings us to something that feels like a fitting place to land after everything we've wandered through tonight. Because if there's one thread that connects all of these systems, your breathing, your circulation, your kidneys quietly balancing your blood chemistry, your endocrine system sending hormonal messages across your entire body, your vagus nerve toggling between rest and readiness, your immune system remembering every pathogen it's ever encountered, your pain system trying to protect you, your homeostatic machinery keeping everything within livable bounds. If there's one thread that connects all of it, it's this. Your body is not a collection of separate systems. It's one integrated, deeply interconnected living process. We've talked about these systems one at a time tonight, because that's the only way to talk about them. Language is linear. You can only explain one thing and then the next thing. But in your actual body, right now, all of these systems are running simultaneously, talking to each other, influencing each other. Your immune system affects your nervous system. Inflammation in your body changes your mood, your energy levels, your perception of pain. Your nervous system affects your immune system. Stress hormones suppress certain immune functions while enhancing others. Your endocrine system modulates your kidneys. Your kidneys affect your blood pressure. Your blood pressure affects your heart. Your heart's output affects your brain. Your brain regulates your breathing. Your breathing affects your blood, pH. Your blood, pH, affects your enzyme function. Your enzyme function affects every metabolic process in every cell. It's all one conversation, happening in real time, right now, in you. And the history of understanding the body has been, in many ways, the slow realization of this interconnectedness. The early anatomists, Vesalius, Harvey, they mapped the parts. The physiologists of the 19th century, Bernard, Ludwig, Starling, they figured out how the parts worked. The 20th century brought the molecular revolution. Hormones, neurotransmitters, receptors, genes, the whole dazzling machinery of cellular communication. And now, in the 21st century, we're increasingly grappling with the systems level question. How does it all fit together? How do you model something this complex? How do you understand a system where everything affects everything else? This is where modern biology starts to look a lot like engineering, actually. Systems biology, as it's called, borrows tools from control theory, network analysis, computational modeling. The same mathematical frameworks that engineers use to understand complex systems like power grids or telecommunications networks. And there's a beautiful irony in that, because for centuries, engineers have been borrowing from biology. The Wright brothers studied bird flight. Velcro was inspired by burrs sticking to a dog's fur. Neural networks in artificial intelligence are loosely modeled on the brain. And now, biology is borrowing back, using engineering frameworks to understand the very systems that inspired those frameworks in the first place. But even with all our modern tools, the body retains its capacity to astonish. There are still fundamental questions we can't fully answer. Consciousness. How does subjective experience arise from neurons firing? We don't know. Not really. We can map brain activity. We can identify correlates of consciousness. But the hard problem, why there is something it is like to be you, remains one of the deepest unsolved problems in science. Sleep itself, the state you may be settling into right now, is still not fully understood. We know it's essential. We know that prolonged sleep deprivation in animal studies is fatal. We know that during sleep, your brain clears metabolic waste through the glymphatic system, consolidates memories, repairs cellular damage. But why evolution settled on this particular solution, this daily period of near total vulnerability and unconsciousness, is still debated. It must be doing something incredibly important, because every animal with a nervous system does it, despite the enormous survival cost of being unconscious for hours at a time. So here you are, lying in the dark, and your body is doing all of this. Right now, your diaphragm is contracting and relaxing, pulling air into lungs where oxygen crosses a membrane thinner than a soap bubble. Your heart is beating. It's been beating since before you were born, and it will beat roughly another 2.5 billion times if you're lucky. Your kidneys are filtering your blood, adjusting sodium and potassium and water with a precision that would make any chemical engineer envious. Your immune cells are patrolling, surveilling, remembering. Your hypothalamus is monitoring your temperature, your hunger, your thirst, your sleepiness, making tiny adjustments you'll never notice. Your bones are slowly remodeling. Your gut bacteria, trillions of them, are metabolizing, signaling, interacting with your immune system in ways we're only beginning to understand. And you don't have to think about any of it. That's maybe the most remarkable thing of all. This entire concert of activity, this staggering complexity, runs without your conscious input. You can choose to take a deep breath, sure, but you can't choose to stop your heart, or tell your kidneys to reabsorb more bicarbonate, or instruct your T cells to stand down. The body handles it. It's been handling it since long before anyone understood how any of it worked, since long before Galen or Harvey or Bernard or Cannon or any of the brilliant, stubborn, curious people whose stories we've followed tonight. They spent their lives trying to understand what your body does effortlessly. And every answer they found opened up 10 new questions, which is how science always works, and maybe how it should work. The mystery doesn't shrink as you learn more. It deepens. You understand more, and in understanding more, you realize how much more there is to understand. But you don't need to understand it tonight. Your body doesn't need you to understand it. It just needs you to rest, to let the parasympathetic system, the vagus nerve, that wandering, ancient nerve, to let it do its quiet work. Heart rate slowing, breathing deepening, muscles releasing their tension, digestion resuming its gentle rhythm, melatonin rising, cortisol falling, the whole orchestra shifting into its nighttime arrangement. You are, in this moment, a living testament to 4 billion years of biological problem solving. Every system we talked about tonight, every feedback loop, Every receptor and enzyme and membrane channel exists because it helped some ancestor survive long enough to pass it on. You carry all of that history in every cell. And right now, all of that history is working together to do something beautifully simple: to let you sleep. So let it. Let the systems do what they do. You've learned a little about how they work tonight, and maybe that's enough. Maybe it's more than enough. The rest can wait until morning, when your cortisol will rise with the light and your body will gently bring you back. It always does. It's very good at this. It's had a lot of practice.